I'm Carl Lejouet, a professor in psychological sciences and your provost. The Provost Distinguished Speaker Series is an annual event where we invite our recently appointed Board of Trustee distinguished professors and endowed chairs to share their scholarship with us. We're really pleased to share this opportunity to bring our community together in appreciation and celebration of the exceptional work we do at UConn. And, seriously, as the pandemic drags on, it just becomes more and more important for us to find ways to do this. I want to thank Dr. Dan Fabris, the Harold S. Schwenk Senior Distinguished Chair in Chemistry in CLAS, for sharing a scholarship with us today. He's newly named to the Schwenk Endowed Chair position, and he actually shared that he's been with us about 2 more months than COVID, so at least he had an opportunity to get a little bit of a sense of UConn and our community early on. His talk today is When Chemistry Meets Biology, New Technologies for Cracking the Epitranscriptomics Code. An endowed faculty position is one of the highest academic awards that a university can bestow on a faculty member. It's both an honor for the named faculty appointee and an enduring tribute to the donor who establishes it. There are currently over 120 endowed faculty positions established at UConn. Alumni and friends of UConn may establish endowed funds in memory of a loved one or in honor of someone who has inspired them. Corporate donors also support endowed positions as a means of giving back to the institution that have developed talent and innovative solutions to problems that provide a competitive edge in the marketplace. And I see our president, Radenka Marek, is here, and she's talked quite a bit about the importance of us really ramping up our philanthropy, and endowed professorships are one of the most important ways to do that. The Harold S. Schwenk Senior Distinguished Chair in Chemistry specifically was established in 1996 by Harold and Paula Schwenk, and I'd like to take a moment to acknowledge both Harold and Paula. We can have a round of applause.
Thank you.
Harold and Paula have been long-standing donors to UConn and class and are recognized as members of UConn's Founders Society. Paula is a proud UConn alum, having received her master's degree from NEAG School of Education. Harold also has strong ties to UConn, having served as a member of the UConn Foundation Board. They were recognized in 1998 with the Honorary Alumni Award. The chair was established in memory of Harold's father, Harold Sr., who received a BS in horticulture from UConn's College of Agriculture, Health, and Natural Resources, and later served as a chemistry professor from 1922 to 1951. At the time, the gift was the largest single gift from an individual donor to the university. Thanks to their generosity, UConn has attracted distinguished faculty like Dr. Fabris in the Department of Chemistry, who serve as leaders in the fields of teaching and research, encouraging collaborative learning and driving innovation both domestically and internationally. To tell you more about the accomplishments of Dr. Fabris, I'm going to turn to my colleague, Dr. Julie Wade, Dean of CLAS. Before I do, I always like to say a little bit about our deans. Now, because Julie is the Dean of CLAS, which is the largest here at UConn, I've already had a chance to talk about her scholarship. And so today I just want to say, being the Dean of CLAS is quite possibly the hardest job in any university. There are so many units. They have such a distinct flavor and a disciplinary set of unique strengths. And one thing I think that is particularly hard being in that role is having to tell the big picture story of where the college is going and why that is important, but also really being able to pay attention to detail, to understand the importance of strong and equitable policy, and really being someone that folks can trust, who is going to approach everything in the same measured, thoughtful way. It is really hard to do, but a college runs just so much better when someone does that. And I will say in my, many years now as an administrator, I have really never met anyone who I think cares as deeply and is as focused as Julie is on creating a set of structures and policies and supports within a college that's so big to truly ensure that, again, that we're being equitable and treating our faculty, staff, and students in a way where they can thrive. And, it's one of those things that maybe can be overlooked, but she's truly great at that. So with that, I will turn it over to Julie.
Thanks, Carl, and really thank you for that lovely introduction. That's really very kind of you. So I have the pleasure and honor of introducing our speaker. I first want to say hello and thank everyone for joining us virtually today. And I want to echo Carl's words and welcome Harold and Paula Schwenk, who established the Harold S. Schwenk Senior Distinguished Chair in Chemistry. It really means so much to the college and to the university that supporters like you come forward to enhance our research enterprise. So on behalf of the chemistry department and myself and the college, thank you. We really are very grateful. So it really is an honor to introduce our speaker today. And I will say, just in preparing these remarks, I've really learned so much about the importance of Dr. Fabris's work. So as Carl already mentioned, his work is a wonderful example of interdisciplinary collaboration, which is something we really focus on in the College of Liberal Arts and Sciences. So the work sits at the intersection of chemistry and biology, which just happen to be two of the largest groups of faculty in the college. And the scholarship focuses on RNA viruses, including most recently the virus that causes COVID-19. Dr. Fabris received his PhD at the University of Padova in Italy, and he was a postdoctoral associate at the National Research Council in Padova, and also at the University of Maryland, Baltimore County. After UMBC, he was on the faculty of SUNY Albany before joining us here, and I am grateful and proud that Dr. Fabris now calls UConn home. He's authored nearly 100 journal publications and more than 200 conference proceedings, and he's earned an impressive $11 million in external research funding for his work so far. Hopefully that will continue long into the future. So please join me in welcoming him for the inaugural Harold S. Schwenk Senior Distinguished Chair in Chemistry, Dan Fabris.
Hey, thank you for the kind words, and thank you for having me. It's a wonderful opportunity to talk about what I'm doing, and I would like to also thank Harold and Paula Schwenk for the generous gift that they did to the university. They gave me the opportunity to come here and start to build a lab and friends here at UConn. So without further ado, I'm going to share my screen with my presentation here. I hope you can see it. Okay, somebody can give me a thumbs up. Okay, so the type of work that I do is really at the interface between chemistry and biology. I'm physically located in a chemistry building, but at UConn, we are lucky that we have all our friends that work in biology that are very close, very nearby. So I hope I can make my way to their building as often as I can. And this talk today is actually more on the biological side than it is on the chemistry side. Again, with the idea of showing what I'm doing going forward, hoping to make some friends, to make some good collaborators here. The type of work that I do is all based on mass spectrometry. Mass spectrometry is my analytical platform of choice. I develop technologies based on mass spectrometry to investigate everything that relates to RNA. I've been working in the past on RNA-protein interactions, RNA ligand interactions. A lot of the work that I do is very much biophysical in nature, structural determination, looking for ligands, looking for possible drug inhibitors. But in the last 10 years, I also became interested in these RNA modification and epitranscriptomics, which is very much biological in nature. And today I will be covering this part of my research program. So why should we focus on RNA? Why is RNA so interesting? Well, RNA can be a potential therapeutic target. So if we look at this pie chart, this is give us an idea of the entire information that is encoded in the human genome. We know that there are 3 billion nucleotides in there. And out of these 3 billion nucleotides, only 2% are actually coding for proteins. I'm going to start a laser. Yeah. So only 2% code for proteins. And these proteins are the target to over 10 to the 4 drugs. So there are more than 10,000 drugs that have been developed and approved. And these are targeting only 2% of the information in the human genome. At the same time, at least 70% of this information is transcribed into RNA. And these RNA used to be called junk because we didn't know exactly what it was doing. And I'm going to talk more about this. Up to this point, fewer than 10 drugs have been developed that target these RNA sequences that are provided by the vast majority of the human genome. So you can already look at this and think about the potential that is there. There is a universe of possible targets that we are still barely discovering these days. As you might be aware, RNA can also be a therapeutic agent in its own right. And the success of mRNA vaccines to control the COVID pandemic, it's actually the pinnacle of these technologies. But there are others that are in the pipeline where RNA is really used as a possible therapeutic. And I would like to venture that at one point, RNA might become a diagnostic feature. And I hope I can demonstrate some of these today during my talk. So a little bit more about the regulation of this information going from DNA to RNA to protein. For the people that are not in the life sciences, I hope you can remember your biology high school. This is called the central dogma of biology. And we can see immediately that the proteins do most of the work in terms of regulating their own expression. And this was true up to until maybe 15, 20 years ago. Certainly, it was true when I was in school. But recently, we discovered that RNA is actually capable of silencing itself. So RNA is capable of regulating its own and therefore regulating the possible expression of proteins. Even more recently, we now discover that proteins are capable of interacting with this non-coding RNA. And this is what we call epitranscriptomic right here. So today I'm going to talk about the potential for these RNA modifications to regulate its own translation into a protein. What are we talking about when we talk about RNA post-transcriptional modification? We are all familiar with the fact that there are 4 canonical bases for RNA. And once these are incorporated into an RNA, there are enzymes that establish some covalent modifications. And the covalent modifications can basically be attached to any of these positions in the canonical bases. You should know that up to this point, the community has discovered over 170 variants. And these are just, as I say, variations of the canonical nucleotides that are established by enzymes. And what do these modifications do in the context of an RNA? Well, we know that RNA can make hydrogen bonds, can recognize proteins. We also discovered that RNA can recognize metabolites. But once a base pair like this has to contend with a modification, it starts to weaken, it starts to change the binding properties, and in most cases, it's also changing the higher order structure of the RNA. So this may be a way to interfere with function just by simply applying some of these covalent modifications into the RNA. And indeed, these modifications are very dynamic in nature, and we are already discovering they can act as on-off signals. Imagine we have an RNA here that can be now modified by an enzyme. We call these writers because they're establishing the modification. There it is. And there are also erasers. There are enzymes that are capable of removing these modification to restore the original RNA or to generate other modified RNA downstream here. In addition to erasers and writers, there are also what they're called readers. There are proteins that recognize the presence of a modification on an RNA strand, and then they recruit other proteins to really start a communication pathway. So how can we detect this modification to study them? Well, very popular these days is this the type of analysis based on next generation sequencing. This provides the sequence of nucleic acid. In this case, there are at least 15 antibodies that are available for 15 types of modification. The workflow is very straightforward, at least on paper. We start from an analyte that might be modified in here. We apply some shearing or some method to cleave this RNA in smaller pieces, and only a few of these pieces will contain the modification that we can now enrich by using these specific antibodies. Once we have a sample of enriched antibodies, now we can really sequence the strands, and by aligning the sequences, that there is software that allows us to identify the position of these modifications. This is very powerful, can work at the entire transcriptome level. We can get a ton of information from there. In addition to these direct enrichment methods, there are also other methods that employ a chemical treatment. So in this case, what we can do is we can divide the initial RNA into aliquots. One aliquot is submitted immediately to the sequencing process that I show you up here. The other aliquot can be treated to modify the complexion of this modification. You see it's changing color from blue to green. And now I can sequence these. And just by comparing the two sequences, chances are that there will be some different nucleotides that will pair to these, different from the normal nucleotide that will pair to the canonical base. And by just comparing these point mutations, we can now recognize the presence of the modification. So it turns out that there is some overlap between these methods. These days, we can go after 23 of these RNA post-transcriptional modifications. As I told you earlier, though, there are 170. The only way that we can analyze all 170 is by using mass spectrometry, which is the technique that I'm using in the lab. And this is due to the fact that we can take advantage of unique masses. Every time there is a modification, the canonical nucleotide will change mass. In addition to that, we can also apply fragmentation in the gas phase, and we will obtain some fragmentation signatures that are different between one modification and the other. So a possible workflow that we've been working on in the lab is we can isolate the RNA. Sometimes we extract the entire RNA from a cell, so we do a global analysis. In this case, we start from the RNA, we digest it using an enzyme into the mononucleotide components. And you see here there are some canonical and some are modified. And then we just do the mass spec analysis that I told you earlier. At this point here, we lose the sequence context because we don't have the strand, the intact strand anymore. But we can do a survey, a very rapid survey, and immediately recognize whether any of these modifications is turned on or off based on conditions. I will show you later some examples. Another type of analysis that we do in the lab is we apply a different type of enzyme. Instead of cleaving everything down to the individual components, now we end up with some stretches of RNA, some oligonucleotides that we can now weigh by mass spectrometry. And already just by mass mapping, by getting the masses of these oligos, we can already start to tell if any of those contain a modification, because again, there will be a shift in mass from what we can predict for the naked sequence. And once we have these maps, now we can submit each of these individual species to tandem sequencing in the gas phase by basically activating the fragmentation in the gas phase. So by doing this process here, now we can also identify the sequence location for these modifications. I'm not going to talk about this today. I will just keep it straight to this survey type of analysis. you should know that in the lab, we multiplex this type of analysis. So we can use a liquid handler where we can do these sample prep steps in parallel on 96 well plates. And then we can run these overnight in an automated fashion in one of the two instruments that we have in the lab at this point. So these allow us to increase considerably the sample throughput and do enough analysis to support the statistics. We need to do biological replicates to be able to quantify these modifications, compare their amounts, I will show you in a moment, and have confidence in what we see in this analysis. You should also know that at the back end, we need software to do this interpretation. At one point, I started to write software myself, and I also started a startup to commercialize this type of software that I devised now with the help from some collaborators. Again, to be able to interpret this data and make progress as quickly as we can. Okay, finally, this is how the data look like at the end. They look like heat maps. Notice here in the first column, I have the names of the modifications. In this case, I'm not separating the modifications according to the position of these modifiers. I'm just using groups that have the same elemental composition, so they share the same mass. And just to show you what type of the samples I'm looking at here, in this case, I'm looking at a cell culture. It's a human liver cell culture, HUH7. This is the control cell, the control set of cells. Now, the same cells were infected with Zika virus. If you remember, a few years ago, there was an outbreak of Zika, and we started to work on that. And we compare what we observed. Notice the modifications now that we observe from the total RNA of the same cells infected with Zika. There are some new modifications that appear, some that disappear. And we compare these patterns with the patterns that we obtain from infecting the same type of cells with dengue virus, hepatitis C virus, and poliovirus. You notice immediately that these patterns, the presence or absence of these modifications, but also their amounts that is conveyed by this color gradient, is different from one another. So these panels of modifications, there are 47 in the control, 44, 41, 40, and so on. So these large number of modifications in the different samples can really identify uniquely these samples. So when we started to look at this data, the first approach that we use to interpret them, we use Venn diagrams. We try to figure out where the common modifications are and where the unique modifications are. Notice here there are 35 modifications that are in common between all the samples. And that's understandable because we're using the same background of these HUH7 cells. But then the different viruses, they already put their own complexion in there. Depending on the type of virus, we have different type of modifications, some that are very unique to the viruses. So already from these early data, we obtained these maybe 5 years ago, we were already thinking about maybe we can use this type of approach as a diagnostic. Maybe we can take samples from patients that are infected by the different viruses and tell them apart. Well, this type of Venn diagram approach is a little bit limiting, though, because it's only using the absence or the presence of a certain modification. These are binary type of information. And instead, our technique, as I said, with these color gradients, is giving us a quantitative abundance. It's giving us the amount of the different modifications that we detected in there. So we really wanted to take advantage of these to make our diversification, our identification of these samples more robust. And we tried many different approaches until we finally decided to use machine learning. Machine learning is ideal to deal with a multidimensional problem. If you think about what are these dimensions of this multidimensional problem, the number of rPTMs in the 40s appear, their abundance, it's another dimension. The number of replicates that we need to do, typically we do at least 5 biological and 5 technical per each biological. So we try to use at least 25 replicates per experiment. And of course, the number of classes. In this case, I was trying to differentiate only 5 type of classes, but later on I will show you that we can increase the number of classes that we want to differentiate from one another. So we started to work with machine learning, in particular with a supervised type of learning. So the way it works, just very quick, we first collect a large data set, then the program randomly split the data set in a 70% and 30% proportion. The 70% portion of this data is used to train a model, a classifier. So each of these, you can see here, is a sample that contains features. And of course, we know the origin of the sample, so we know the class, the identity, basically, of this sample. And we just submit them to the program, and the program works in an iterative way to build a classifier, to build a model that will take in account all the commonalities and the differences between the samples to generate a set of rules that allow us to then identify what can be an unknown sample. The process, as I said, works in a very iterative fashion. In this case, we use a leave one out cross-validation. You see all these samples here. The first sample is left out, and all the others are used to generate a model. And at the end, this sample that was left out is used to test the validity of the model. This is the first round of guessing, if you wish. The second round of guessing is done by leaving out the second sample, and all the remaining samples are used to generate the model, and then this sample is used to test again. And this is done for all the possible samples. So at the end of the process, we are able to distill a very rigorous set of rules, very stringent, that allow us now to assign classes to a sample that might not have a label. So once we generate the model, the classifier like this, we obtain a matrix like this. This matrix gives us an idea of the learning. Was the learning any good? Well, I should explain how it works. On the y-axis, we have the actual samples. There are multiple samples here. They're just identified by this bracket in there. Instead, on the y-axis, we have the names of the classes in here. So in this case, I compare a sample that was the mock cell line that I've been using, the Zika infected, the HCV, the dengue, the PV infected, but I also threw in a set of samples from Aedes aegypti. Aedes aegypti is the host for Zika, and you should know that I don't have the map here, but the distribution, the natural habitat of this mosquito is actually extending to North America. Okay, you know that the outbreak reached Florida at one point, but it was mostly due to travelers. But actually, the mosquito live in Florida. So that's why we were interested in looking at these mosquitoes, and we also look at the mosquitoes infected with the Zika, right there. And again, this is just the learning process. This is just telling me the most samples are assigned 100% with 100% accuracy to the proper class. This line here that you see is a sample of low quality. We have a way to recognize samples that are low quality. And in this case here, where a sample was assigned to multiple different classes, we typically chalk this up to possible contamination of the samples. So if we're not careful in how we collect and handle the samples, this type of treatment will also raise a flag on them. So remember, this work was done on only 70% of the data set. The other 30% of the data set was actually to test the accuracy of the model. So once we build the model, now we're going to use the remaining samples here. But in this case, we submit them as unlabeled. We don't tell the program the name, the identity of these samples, and we just see if on its own account, the classifier is able to assign the correct name of the class to these samples. So also in this case, we end up with a matrix IDs. Again, the samples are listed here, and now I listed them with their actual name. And these instead are the classes that were assigned by the program, by the classifier. And notice here, the accuracy is 100% almost across the board. There's only a few samples here that were misassigned, but still we were up close to 90% accuracy. Even for the most complex samples, I tend to compare an entire RNA panel from a mosquito and the same mosquito infected with Zika. Okay, so we were very, very happy with these. We started to do more work in these, and now we really started to use these classifiers to go after true unknowns, samples that we collected from the field without knowing what it is, and having a collection of classifiers for many different type of samples. We've been testing the ability to recognize these unknowns, which we hope will be the diagnostic application for this type of treatment. In this case, I'm just showing you again a testing set here, just to give an idea of the capabilities of this classifier. In this case, I'm comparing Salmonella typhimurium with Klebsiella aerogenes. This is cause for several infections. This is actually involving sepsis. I'm very interested in using the technique to detect sepsis in a patient in the emergency room. These are different serotypes of E. coli. One of these E. coli is a benign strain that we use in the lab for molecular biology applications, but the other one is actually producing a Shigella toxin. This is Streptococcus pneumonia, Listeria. There's a few human cell lines in here because we wanted to compare them to one another. You see, even though they all share the same genome, they're all human cell lines, we can differentiate them using the technique. We can differentiate them from blood, and also from blood containing a drug-resistant strain of E. coli. So these are all possible applications that are encapsulated in one single slide in here. And up to this point, we look at cell cultures, like I show here, but also surgery specimens, biopsies, blood, saliva, all sorts of biological fluids, again, to test the potential for this application to identify pathogens in any of these materials. I'm already in advanced talks with possible collaborators to start to look at orthopedic samples, to some brain tissues from mouse, and maybe to some ticks. Wouldn't it be wonderful to be able to grind a tick and do this type of analysis, stack the RNA, do this analysis, and immediately after 1 hour be able to tell if this tick carries Borrelia, who is the agent of Lyme disease. So these are the possible applications in the diagnostic field. But there is also a lot of potential here to support biomedical research. So this is another project that we're involved now, and we are looking at possible interactions between HIV and drugs of abuse. So you're by now familiar with this pandemic. That's also a pandemic of HIV infections. HIV has become now a chronic disease. It was not when I was a student. When I was a student, being HIV positive was almost a death sentence. But now patients can take these cocktails of inhibitors, and they can survive for many, many years without any problems. So it turns out there are 38 million people living with HIV at any given time. And in the world, there are also 16 million people that are drug users, injection drug users. These numbers are staggering. Every time I look at these, I weep. There is also an overlap between these two groups. There are at least 3 million people that are HIV positive and are also using injection drugs. So it turns out that these people are at very high risk because drug of abuse can hasten the progression from HIV positive to a full-blown AIDS. So even if these patients are compliant with the cocktails, they can still develop full-fledged AIDS if they're also taking drugs. So we believe that we have an hypothesis. We're not the only one that are now studying this. And we believe that the hypothesis of how HIV hastens the progression to full-blown AIDS is through RNA modifications. The community is finding now that there are several modifications that are involved in HIV, and I will show in a second also in some of the signaling, neuromodulator signaling in the brain. I'm just using M6A, which is the most famous, the most studied, because we have some antibodies available for these, so we can use next generation sequencing to get information. And over time, people have found that these modifications of M6A can prevent the degradation of viral RNA and the degradation of the mRNA that is used to make viral proteins. It can also modulate the splicing, can enhance the transport across the nuclear membrane, and in general enhances the viral gene expression. So every time these M6A is stimulated, we see these effects that are all enhancing on viral replication. At the same time, in the central nervous system, the M6A is involved in the dopamine signaling. You know, dopamine is the reward mechanism, and a lot of these drugs of abuse work by activating the dopamine, the reward mechanism. In particular, in the case of M6A, there is a writer enzyme that can get activated to make M6A, and then there is an eraser enzyme, the FTO, that modify this modification into a metabolite. It's unclear if this also has some signaling properties. So it turns out that when the writer enzyme is silenced, the amount of M6A in the brain decreases, and this will also cause a decrease in dopamine signaling and a decrease in learning. You should know that dopamine also works in memory and in learning processes. When the eraser enzyme now is silenced, the M6A goes up. Actually, it turns out that in some of these signaling pathways, not in all of them, the activity of this dopamine receptor decreases, and also the dopamine synaptic release decreases. Okay, so some of these seem a contradiction with one another, but it's really these effects are operational in different regions of the brain. That's why we need to be careful when we look at this. But the take-home message for us is that this M6A is also heavily involved in a neuromodulator signaling. And as I said, this is just one of 170 possible modifications. So our hypothesis here that we're investigating is that these modifications can really work as communication hubs. So the drugs of abuse activate, in this case, dopamine signaling. This will increase regulation, not just of M6A, but of a series of different modifications. And in turn, these will affect the HIV replication. So we started to do work in this. I just give you one quick example of experiment. We have a ton of data that we are trying to rationalize right now. One experiment that we did was we've been working on an immortalized cell line of astrocytes, astrocytoma, and because we can propagate them in vitro rather easily. So our approach is first we're going to learn as much as we can in vitro, and then we will go to primary cells, and then eventually in animal models. And the idea here, we started the infection of the cells. We waited for at least 12 hours to have at least a cycle of replication. And then we added dopamine. And after we added dopamine, we started to take aliquots after 1 minute, 5 minutes, 2 minutes, 6 hours, 12 hours, 24 hours. We really wanted to see a time course here because we're aware of the fact that neuromodulator, once they're applied to a cell, they have a time course. There may be reuptake, there may be a lot of different processes going on. We wanted to have a temporal picture of what was going on. So we did this by adding the dopamine after 12 hours from infection, or adding it after 24 hours post-infection. And again, taking the aliquots at different points. I'm not going to show you all the data that we have, just a representative case. In this case, these are the monomethyl okay? And in this graph here, we are subtracting the abundance of these cytidine in the sample. We are subtracting the abundance of the control from what we observe for the different type of samples. So we did a control astrocytoma treated with dopamine. We did another experiment where we just infected with HIV, and then we did both, like I showed you earlier. First, we infected with HIV, and after 12 hours or 24 hours, we added the dopamine. So immediately, we see that as compared to the control is here, right? Because we're doing the difference, this value will be 0. When we add dopamine, we see that these modifications are down-regulated. See, the signals are going down, they're going negative. Instead, in the case of HIV, they're going positive. And if we have both HIV and dopamine, they're actually even more upregulated in here. And you see the same trend here. Actually, after 2 hours from dopamine, we see a very high abundance of these monomethyl we have a list. We have at least 5 excuse me, 5 or 6 types of modifications with similar trends. Excuse me. Okay, so I'm getting to the home stretch here. How do we investigate now this relationship? How do we figure out the mechanism? Now what we see is just a phenomenon. We have these cells that are treated with a neuromodulator that is mimicking these drugs of abuse. We have the data from infected HIV-infected cells. Now we need to start to put things together and figure out the mechanism. One possible approach is we've been using bioinformatics. We've been using Cytoscape with a relational database that searches all possible interactomic databases, trying to find the relationship between genes. So by using these, we generated maps that connect the dopamine receptors. In this case, this is the dopamine receptor D2, and it's connected through different genes to the biogenetic enzymes of these modifications. You see this NAT10, for example, takes a normal RNA and modifies it, and maybe this was an active RNA, and now this becomes an inactive RNA. In the case of the monomethyl cytosine, this might be an example. This is the biogenetic enzyme that makes M5C, starting from the regular C, and this might be doing the same process of deactivation or activation, but these are all under control of a receptor, not only under control of a receptor, but also under control of some genes that are active during HIV replication. You see these TAR, BP1, these HTAT, IP2. These are all part of the response of the host cell to the HIV infection. And in here, I'm also adding some genes that are part of normal host processes where they do RNA metabolism. So you can already start to see the connectivities here. And what we are doing now is we are starting to silence one at a time to reconstruct these pathways and then observe what happens to the RNA. Because this will give us the mechanism of action and will tell us how the different pathways talk to one another. So with that, I think I'm at the end of my time. I just want to summarize what I show you. I showed that mass spectrometry enabled the global and the targeted analysis of RNA modifications. I didn't show you really the targeted, but we have ways to pull down a specific RNA instead of doing this global analysis. All the data that I showed today were from global analysis, just grinding down the cells, extracting the RNA, and do the workflow that I showed you earlier with a robot. The beauty is that now these panels of RNA modifications can really be differentiating features. I showed that machine learning can really take advantage of all these modifications to come up with very stringent classifiers. We can really differentiate cells that contains pathogen. And that's why we think that this technology has an excellent potential for diagnostic application, but also for basic biomedical research, which is something that we will continue to do to deepen our knowledge of this epitranscriptomic regulation. And with that, I would like to stop here by thanking Harold and Paula Schwenk again for the opportunity to be here and do this work here. These are the current members of the lab here at UConn. Most of the work that you saw today was the work by a talented graduate student, Limin Deng, who is actually going to defend next week. So I'm very happy to say that. These are the other students. We were able to recruit one undergrad in 2 years. That for me, it's very dismal. I'm really trying to recruit more undergrads. Hopefully, when there is more traffic in the building, we will be able to capture more people. And here I had other graduate students and postdocs in the lab. Former members that did a lot of work in this project were Rebecca Rose here and Will McIntyre. Collaborators here, I have a long list of collaborators. I had to put the dots in there. You probably realize that the type of technology that I'm working with are very, they can be multi-use. And that's why I always end up working with a lot of good people. Current support, I enjoy general support from NIH because of the biomedical nature of these applications. And this is my my employee in the startup, Ribodynamics. And I also have to thank him for the help he gave us in developing this software. And with this, I will just stop here, and I will be happy to take any question that you might have.
Thank you very much, Professor Fabris. This is Jeffrey Schulson. I'm stepping in for my colleague, Michael Bradford, who was unable to participate, and to serve as moderator for any questions and answers. Maybe you should take your screen, your share down so we can see each other.
Yeah, that's a good idea.
I'm happy to direct any questions your way. If you want to indicate you have a question either by using the hand raise or just by putting your name in the chat, that would be great.
I'm trying to browse through the names, see if anybody has a...
Well, since you mentioned your efforts to try to recruit some undergrads, and since I have absolutely no background in biochemistry to draw upon for asking a meaningful question on that field, let me ask you about what you're looking for in undergraduate. What kinds of undergraduates are you interested in recruiting, and what kind of work would they be doing for you?
Yeah, so you saw that it's very broad, the type of work that I do. And I'm looking for any undergrad in chemistry, biology, any life science undergrad would be welcome. I typically prefer to get them young, to get them freshman, sophomore, because then I have the chance to work with them. My students, my postdoc can work with them. We can really shape them in a sense. And it's easier when they're still very, very open, even if they don't have the fundamentals, they're gonna learn the fundamentals as they go. I would love to be able to recruit students from engineering, from computer science. I've been looking forever for students that can do some coding. And because most of people that I know that do coding, they're very interested in other topics, and instead there might be a career for them in bioinformatics. So I'm always trying to use this as a selling point when I talk to people. Listen, you may be good at coding, but you can apply your capabilities to support biology, to support biological application, and there is a market there. And again, not too much luck, but I think it's not because we don't have the good students. I think the problem is still the exposure. You know, being able to get face, my work in front of them. But these are the type of people that I would love to have.
Thank you. I don't know if you've noticed, but the director of our honors program, Dr. Jennifer Lees-Butts, has indicated that she's going to try to connect you with some of her students and with the Office of Undergraduate Research. I'm sure you'll be able to find some.
Thank you very much.
There.
That's very appreciated.
Yeah, I mean, it is a bit of a handicap not to be on campus physically with everyone and be able to kind of just have those occasional encounters that would allow for those opportunities. Please let me invite my colleagues who are on the screen to share any questions or observations they might have.
I'm
Yes,
also trying.
I see President Merrick has raised her hand. Let me ask her to go ahead.
Hey, Daniel, first, I want to say how happy and thrilled we are to have you at UConn.
Thank you.
I like your broad perspective and linking the biology and chemistry and everything that you do. I think there is no large issues in today's world that can be solved by one discipline, and you crossing those disciplines shows how we have to think. I think there are many opportunities. What I learned the most from students when we move them in the houses. And that's where they says, I'm looking for this professor, I'm looking for this. And I get many undergraduate students. Pamir Alpay is now our interim vice president for research. And I think it would be beneficial on posting on the web page and asking our faculty who are looking for undergraduate and giving small scholarship or something, because I could see that when we had the opportunity, when we provided it 5,000 scholarship, how many students applied. And in 5 days, in 1 week or 5 working days, we got 150 applications. So I think we have experienced how to recruit, but it has to be cool science. It has to be something appealing to them, and we have to think out of the box. So I think if you approach Pamir and tell him what is the idea, and if there is any funding that you can support or Dean can support, or you can support from your startup, I'm sure you will attract many brilliant students. The way how I attract them, I do the independent study. So I provide them the credit, or I paid like a co-op or interns. So there are different models, but we definitely want you to be successful at UConn. We want you that you bring the students both from chemistry and biology, and they work together, because that will be a great opportunity for them to learn.
Thank you for the suggestions. I'll take them to heart. I'll do it.
Thank you. There's an enthusiastic endorsement of what President Merrick said in the chat about scholarship and credit making a dramatic difference. Other comments, questions, observations? I mean, as a complete novice and ignoramus on these things, I'd love to ask you a little bit about. You alluded to this as possibly also having potential for diagnosing or sensing things like the COVID virus. Have you explored that at all in some of your work as well at this point?
Yeah. So at this point, I'm using polio as a model for a virus. My lab is a BSL-2. You know, with these viruses, you need to do work under biosafety. You know, follow the biosafety guidelines. And the SARS-CoV-2 is a BSL-3. So what I've been doing is I've been sharpening my tools. Basically, I'm using polio to refine the technique, and I'm one step away from being able to prove that we get the sensitivity necessary to be able to detect the virus in the blood of an infected patient. So if I can prove that I can do polio, then I hope I can make some friends in the medical school where they're really working with the virus and have them work with me, because they will have to do the sample prep again in their lab, in a BSL-3 lab, and then provide non-infectious, after digestion, after the treatment, the samples will be non-infectious. At that point, we can do the analysis here. Again, for safety reasons. But again, the only way to do this type of work is really by collaborating. There is no way around it.
Right.
Luckily, polio, we are all vaccinated for polio.
Yeah.
At least
we hope so.
Yeah, I don't remember ever meeting somebody who was not vaccinated.
Yeah, with the sort of anti-vax world growing, who knows where we're headed? Hopefully not in that direction. Well, unless there are any other questions, we're coming to the top of the hour, and I want to thank you, Dr. Fabris, for it seems like extraordinary work to me, and I think what impresses me, among the many things that impress me about the work, is just how collaborative and multidisciplinary it is, and how critical it is to have so many different kinds of investigators involved who can provide their expertise, the drawing from the various life sciences, but also from machine learning and the folks who do data analysis and the coding. I mean, this is really, as President Merrick said, these problems, these are complex problems, and they are not solvable except through complex systemic approaches that engage a variety of disciplines. And your work seems like a model for that. So I'm really grateful for you for sharing your work. And we're delighted that you're at UConn, and we hope that soon enough we'll be able to actually meet in person rather than through screens.
Yeah, thank you for your word, and thank you for everybody for being so kind. And I'm very happy to be here. The feeling is mutual. You should know that. And thank you for everybody who came. If you have any question for me, send me an email. I'm very easy by email. If you want to talk about things, you see opportunity, just get in touch.
It's Harold Schwenk here. I wonder if I could just add one comment.
Of course.
I noticed that President Merrick talked about the young student bodies and the enticement of what I would consider a rather modest remuneration might bring quite a bit of interest. I speak from experience myself on that front, where for reasons that are irrelevant, I had wanted to have part-time employment in my undergraduate school, and one opportunity that was available took me into the field of computer science, which is then where I ended up, having been lured into it by a rather modest remuneration. So I do think that is quite a good incentive and gets some very talented, hardworking people. Now, office space can be a challenge as well if you are lucky enough to get 2 or 3 or 4 potential volunteers or coders, as you say. So I know human resources are one thing, physical resources are another. Hopefully, you have sufficient dollar resources and equipment. So with all of those ingredients, this is very exciting work, and I'm very happy to hear of what you're doing. Thank you.
Thank you. Thank you for your words.
Thank you very much, Mr. Schwenk. I appreciate your comments and, of course, your generosity, your family's generosity.
Yes.
All right. Well, I again, thank you all for joining us. And thank you again, Dr. Fabris, for sharing your work with us. And we'll look forward to seeing you for the next in this series.
Thank you.
Thank you.
Have a great day.